One evening, everything looks perfect. The tomato plants are standing tall, their deep green leaves catching the last light of the setting sun. The fruits are forming, small, round, full of promise. A farmer walks through the rows, brushes a leaf with his hand and smiles. Tomorrow, he thinks, tomorrow they'll be even better. He comes back in the morning. And they're dead. Not wilting, not struggling, dead, collapsed, brown, as if someone reached down in the night and switched off the power to an entire living thing. It happens in hours, sometimes faster. And when it happens for the first time, farmers don't just feel loss, they feel something stranger. They feel confused because nothing warned them. No storm came through, no frost, no visible pest. The soil looks the same. The sky is the same. And yet an entire row of plants, sometimes an entire field, is gone. This is the story of why that happens. And once you understand it, you will never look at a tomato the same way again. Let's start at the very beginning, not with the tomato, with the soil. Most people think soil is just dirt. Brown, dry, unremarkable stuff that sits under our feet and holds things upright. But here is the truth. And this is one of the most important things you can know about where your food comes from. Soil is alive. Not alive in a poetic sense. Actually, biologically, measurably alive. A single teaspoon of healthy garden soil, the amount you could fit on a small spoon, contains more living organisms than there are human beings on this planet. Bacteria, fungi, nematodes, protozoa. Tiny mites and microarthropods that are so small you'd need a microscope to see them, but so numerous they form entire invisible ecosystems beneath every footstep you've ever taken in a garden. These organisms are not random. They have relationships, hierarchies, predators and prey, partnerships so ancient and so efficient that they evolved hundreds of millions of years before the first tomato ever existed. And the tomato plant, when it pushes its first root down into the soil, is not just anchoring itself. It is entering a world full of allies and enemies. The question that most people never ask is this: which one does it meet first? To understand what kills a tomato plant overnight, you have to understand something about the plant itself. Something that surprised even scientists when they began studying it carefully. A tomato plant is not just a passive thing. It is not simply sitting there, waiting to be watered and fed. It is constantly communicating, constantly sensing its environment, constantly making decisions. When a tomato root pushes through the soil, it releases chemical signals, invisible substances that drift through the microscopic spaces between soil particles. These chemicals are a kind of language. They attract beneficial fungi and bacteria. They repel certain insects. They tell neighboring plants something is happening here. Scientists call these root exudates, and they are one of the most fascinating things in all of agriculture. Because what a plant says with its chemistry determines, to a surprising degree, what comes to live in the soil around it. The plant is building its own neighborhood, choosing, in a sense, its own companions. But here is where the story gets dangerous. Some of the organisms in the soil are listening to those same chemical signals, not to help the plant, but to find it. The most important word in the story of overnight tomato death is one that sounds almost too simple for how destructive it is. Fungus. Not just any fungus. Specific species of soil-dwelling fungi that have evolved over millions of years to do one thing very well: destroy the vascular system of a plant from the inside. And they do it quietly, invisibly, while the farmer sleeps. While the sun goes down, and the temperature drops, and the night settles over the field. The most infamous of these is called Fusarium oxysporum. Say that name to any experienced tomato grower, and watch their face change. Fusarium is a species of fungus, or more accurately, a family of related species, that lives in soil on almost every continent on Earth. It is patient. It can lie dormant in dry soil for years, decades in some recorded cases. It does not need a living plant to survive. It feeds on organic matter, persisting quietly, waiting. And when a tomato plant arrives, when those root exudates begin to flow and the chemistry of the soil shifts, Fusarium wakes up. Under a microscope, Fusarium spores are almost beautiful. Tiny, banana-shaped structures called macroconidia, curved. Delicate-looking, almost translucent. You would not look at them and think, this is what kills farms. But the moment a Fusarium spore makes contact with a tomato root, the process begins. And once it begins, it moves faster than most people realize. Here is what happens inside the plant. And this is the part that most people, even people who grow tomatoes all their lives, never fully understand. The Fusarium spore germinates. It begins to grow. It pushes a thin, hair-like filament called a hypha into the root tissue of the tomato plant. This penetration can happen within hours of contact. The plant's root cells, which have receptors designed to recognize certain threats, sometimes fail to detect Fusarium at all. Why? Because Fusarium is extraordinarily good at mimicking signals that the plant recognizes as safe. It is, in the most literal sense, a disguise. The hypha moves inward, through the root cortex, the outer layers of root tissue, and finds its way toward the xylem. Now the xylem is critically important. If you imagine the tomato plant as a building, the xylem is the plumbing. It is the network of tube-like vessels that carry water and dissolved minerals from the roots all the way up to the leaves, the stems, the flowers, the forming fruits. Every part of the plant above the soil depends on the xylem to deliver what it needs to survive. And Fusarium colonizes the xylem. It begins to grow inside these tubes, to multiply, to spread upward through the plant's own transportation system, using the very pathways the plant designed for itself. As it grows, it produces toxins, mycotoxins that attack the surrounding plant cells, and it produces another substance, a thick, gummy material that begins to physically block the flow of water through the xylem tubes. Now picture what that means for the plant above. Water is still in the soil. The roots are still there, but the pathways have been blocked. The water cannot rise, and a tomato plant with its large leaves and its actively growing fruits and its constant need for hydration cannot survive without that flow, not for long. The leaves begin to wilt, but not all at once. Often, the wilting starts on one side of the plant. One branch droops while the other still stands. This is actually a clue, a diagnostic sign that experienced farmers and plant pathologists look for. When wilting is asymmetric, one-sided, starting at the top before spreading down, Fusarium wilt is one of the first suspects. And then, sometimes within a single night, sometimes faster, the entire plant collapses. But Fusarium is not the only culprit, not even close. There is another organism in the soil. Another ancient enemy of the tomato plant that works in a completely different way but produces the same devastating result. A plant that was alive at sunset and dead by dawn. Its name is Phytophthora infestans. If you know your history, that name might ring a distant bell. Because Phytophthora, which belongs to a group called Oomycetes, often called water molds, is the same organism, or a close relative of it, responsible for the Irish potato famine of the 1840s. The blight that killed the potato crops, that caused the starvation of over 1 million people, and drove another million to emigrate from Ireland in a span of just a few years. That catastrophe, which reshaped the population and culture of an entire country, was caused by an organism so small you cannot see it without a microscope. And it is still here, still in the soil, still in the air, waiting. Phytophthora operates differently from Fusarium. Where Fusarium enters through the roots and destroys from the inside, Phytophthora attacks the leaves, the stems, and the fruit from the outside in. And it has one remarkable, terrifying advantage. It loves the same condition that farmers most want for their plants. Warm nights, high humidity, recent rain. The conditions that make a tomato plant grow beautifully are exactly the conditions under which Phytophthora spreads fastest. Let's talk about what happens during a Phytophthora event, because understanding the timeline is part of understanding why it seems so sudden. The organism begins as tiny spores, called sporangia, that are carried through the air, not through water, through air. They can travel on the wind for miles from an infected field or even from a compost pile where diseased material was improperly discarded. When those spores land on a tomato leaf, particularly when that leaf is wet from rain or dew, they germinate. They release swimming cells called zoospores that move through the thin film of water on the leaf surface and find the stomata. Stomata are small pores on the surface of leaves that the plant uses to breathe, to take in carbon dioxide and release oxygen and water vapor. They are essential for the plant's survival, and they are also a door. The zoospores swim through the water film, find a stomatal opening, and enter the leaf. Inside the leaf tissue, Phytophthora grows with alarming speed. It threads through the cells, kills them, produces more spores. Under the right conditions, warm, wet, humid, the time from initial infection to visible symptoms can be as little as 5 to 7 days. But the visible collapse, the moment when the plant seems to die overnight, comes at the end of that process. What looks like sudden death is actually the final stage of a disease that had already been running for days, invisibly, inside the tissue. This is one of the cruelest aspects of plant disease. The warning is hidden, the damage is silent, and by the time you can see it, it is almost always too late. Now some farmers might be reading, or rather, watching this and thinking, but I've grown tomatoes for years and never had this happen. Or maybe you have, and you've been mystified. Both experiences are completely valid, because here is the thing about plant disease. It is not random. It is conditional. There is a concept in plant pathology called the disease triangle. It is one of the most useful ideas in all of agriculture, and it is surprisingly simple. For a plant disease to occur, 3 things must be present at the same time: a susceptible host, a virulent pathogen, and a favorable environment. Remove any one of those 3, and the disease cannot happen. This is why a fusarium-rich soil might host healthy tomatoes for years, until a drought weakens the plants and they become susceptible. It is why a Phytophthora spore in the air does no damage on a dry day, until a week of humid, warm nights gives it the environment it needs. It is why some tomato varieties planted in the same field where others have died remain perfectly healthy, because they are not susceptible to that particular strain of the pathogen. The disease triangle is not just useful for understanding plant disease. It is a framework for understanding almost all of biology. When 3 conditions align, something that seemed impossible becomes inevitable. And tomato farmers, whether they know the name of the concept or not, spend their entire growing season trying to keep those 3 points of the triangle apart. There is another killer, one that doesn't come from fungi or water molds, one that is, in many ways, even harder to detect. It is bacterial. Ralstonia solanacearum is its name. And if Fusarium is the patient, invisible assassin, and Phytophthora is the fast-moving storm, then Ralstonia is something different again. It is arguably the most destructive plant bacterium on Earth. It affects more than 250 different plant species. It kills through the soil, and it can persist in waterways, in rivers, in irrigation channels, in groundwater, for years. Here is how it works. Ralstonia enters a tomato plant almost always through the roots, particularly through wounds. Cracks in the root surface caused by nematodes, insects, or simply the physical pressure of growing through rough soil. Once inside, it does something remarkably similar to what Fusarium does. It colonizes the xylem. It multiplies inside the water-conducting vessels of the plant with extraordinary speed. And then it produces massive quantities of a slimy, gelatinous material that physically clogs those vessels completely. The resulting wilt happens fast, dramatically fast. A tomato plant can go from healthy-looking to completely wilted within a day or two of the bacterial population reaching a critical mass inside the xylem. And in hot weather, above 25 degrees Celsius, the bacteria multiply faster. The disease progresses faster. The death is faster. There is a simple field test that farmers and plant pathologists use to check for Ralstonia. You cut the stem near the base of the plant and hold the cut end in a glass of clear water. If the water turns cloudy, if you can see thin, white, thread-like streams drifting down from the cut stem, that cloudiness is bacterial ooze. It is millions of bacteria flowing out of the xylem with the sap. It is one of those tests that, once you have done it, you never forget. But wait, because we haven't talked yet about the thing that, in home gardens especially, is responsible for more overnight tomato deaths than perhaps anything else. It has nothing to do with bacteria or fungi or water molds. It is far more common, far more preventable, and almost always it is caused by the farmer themselves. Overwatering. This sounds too simple, too mundane. But the physiology of what overwatering does to a tomato plant is anything but simple, and understanding it reveals something extraordinary about how plants actually function. Tomato roots, like all plant roots, need two things from the soil: water and oxygen. We all know about the water, but the oxygen part surprises people, because roots breathe. They take in oxygen from the air spaces between soil particles, spaces that, in a well-structured, healthy soil, make up roughly half the total volume of the soil. When you overwater, those air spaces fill with water. For a short time, this seems harmless. But within hours, the oxygen in the waterlogged soil is consumed by the bacteria and other organisms that are suddenly thriving in the wet conditions. And then the roots are sitting in water with no oxygen. This is called anaerobic conditions. And in anaerobic conditions, the root cells begin to die, not from thirst, from suffocation. Dead root cells cannot absorb water. So in one of nature's most bitter ironies, a plant that has been given too much water ends up dying of dehydration. The leaves wilt exactly as they would in a drought. The stems go limp. The plant collapses. And often the diagnosis is wrong. The farmer sees a wilting plant in soil that is visibly wet and thinks, something must be attacking it, and sometimes something is, but sometimes the cause was a garden hose. This brings us to a concept that changes how you see everything about plant health. The rhizosphere. The rhizosphere is the thin zone of soil immediately surrounding the roots of a plant, typically just a few millimeters in thickness. But within that tiny space, something extraordinary happens. The chemical signals released by the roots create a microclimate so different from the surrounding soil that scientists sometimes describe it as a completely separate ecosystem. In healthy rhizosphere conditions, beneficial organisms compete fiercely with harmful ones. Certain bacteria, notably Bacillus subtilis and related species, produce antifungal compounds that suppress fusarium spores. Certain mycorrhizal fungi, the word mycorrhizal comes from the Greek for fungus and root, form partnerships with plant roots that physically extend the root system's reach, while also providing some protection against pathogens. These beneficial organisms are not just helpful. In a truly healthy soil, they are the plant's immune system. They are the first line of defense that keeps Fusarium and Phytophthora and Ralstonia from establishing themselves in the first place. And here is the thing that modern agriculture is grappling with very seriously. Many of the practices that make farming efficient in the short term, heavy tillage, chemical fertilizers, repeated pesticide applications, damage this invisible immune system. They reduce the diversity and abundance of beneficial organisms in the rhizosphere. And when that protection weakens, the pathogens that were always there, patient and waiting, find it far easier to do what they evolved to do. This is one reason why, in recent decades, there has been a growing movement among both large-scale farmers and home gardeners toward what is called regenerative agriculture, farming practices designed not just to grow crops but to actively rebuild the living community in the soil. Cover crops, reduced tillage, compost, microbial inoculants, these are not just trends. They are, in many ways, a return to the conditions that allowed plants to thrive for hundreds of millions of years before we began removing them. Let's take a moment to talk about something that many tomato growers experience but fewer fully understand: the difference between wilt disease and drought stress. Because from a distance they look identical: drooping leaves, limp stems, a plant that looks defeated. And the instinctive response, both in farming and in home gardening, is to water the plant, which, depending on the cause, is either exactly the right thing or exactly the wrong thing to do. Drought stress wilts a plant uniformly. All the leaves droop together. The soil is visibly dry. The temperature is high. Water the plant in the evening, and by morning, in a healthy plant, the leaves have largely recovered their turgor, their firmness, their upright posture. The plant bounced back because the problem was simply a lack of water, and the plant's vascular system was intact and fully functional. Disease wilt is different. It is often asymmetric, one side more affected than the other. It frequently starts at the top of the plant rather than the bottom, because the pathogen has been ascending through the xylem from the roots upward, and the topmost parts of the plant are farthest from the water source. And critically, disease wilt does not recover when you water. The plant wilts more. It collapses further, because the problem is not the amount of water in the soil but the plant's ability to move that water through its system. This distinction matters enormously, because a farmer who diagnoses disease wilt as drought stress and responds by increasing irrigation is doing exactly what Phytophthora and Ralstonia most want: more moisture, more favorable conditions, more spread. There is another nighttime killer that belongs in this story. One that is neither fungal nor bacterial but animal, and one that most gardeners encounter at some point with a mix of disbelief and dark humor. Cutworms. Cutworms are the larvae of several species of moths in the family Noctuidae. They spend the day underground, curled in a C-shape just below the soil surface, entirely hidden and dormant. And at night, specifically at night, they emerge and feed. Their feeding method is precise and, from a gardening standpoint, almost surgical. They do not eat leaves like many other pests. They eat the stem, at the base, at or just below the soil surface. They chew through the stem entirely, severing the plant from its root system in a single feeding session. And then they disappear back underground before dawn. The farmer comes out in the morning. The plant is lying on its side. The stem is cut clean, not ragged, not diseased-looking, but cut, as if someone came through the garden with a very small, very sharp knife. And next to the fallen plant, sometimes, you can find the cutworm itself if you dig gently in the soil, fat, gray or brown, curled up and sleeping off its meal. This is why the phenomenon has such a memorable name, because that is precisely what it looks like: a plant that was cut. Now let's climb above the individual plant and think about this from a wider perspective. Because all of the things we have talked about, the fungi, the bacteria, the oomycetes, the insects, the roots, the water, do not happen in isolation. They happen within a system. And understanding that system is what separates a farmer who loses crops year after year from one who, over time, learns to work with the land rather than against it. There is a concept called integrated pest management, IPM, that professional growers, agricultural universities, and farming advisors around the world have been developing and refining for decades. The core idea is simple even if the implementation is complex. Rather than reacting to disease and pest problems after they occur, you build a system that makes them less likely to occur in the first place and catches them earlier when they do. IPM for tomatoes, involves several strategies that reinforce each other. The first is crop rotation. Fusarium, Ralstonia, and many other soil-borne pathogens build up in the soil when the same plant family is grown in the same spot year after year. Tomatoes belong to the Solanaceae family, the nightshade family, along with peppers, potatoes, and eggplants. Planting any of these crops in the same soil season after season is, from a pathogen's perspective, a buffet that never closes. By rotating crops, growing tomatoes in a plot one year, then moving to a non-related crop like corn or beans the next, you interrupt the pathogen's life cycle. You force it to wait, and in that waiting, populations decline. The second is resistant varieties. This is one of the greatest achievements of plant breeding in the 20th century. Scientists and breeders have developed tomato varieties that carry specific genetic resistance to particular pathogens. On a seed packet or in a seed catalog, you will often see letters after the variety name. V, F, and T. A. Each letter indicates resistance to a specific disease. V for verticillium wilt, F for fusarium, N for nematodes, T for tobacco mosaic virus, and so on. These are not GMO modifications in most cases. They are the result of traditional breeding programs that identified resistant wild tomato relatives and carefully crossed them with commercial varieties over many generations. Planting a tomato variety that carries F resistance does not mean fusarium isn't in your soil. It almost certainly is. It means the plant you have chosen has a genetic toolkit that allows it to recognize the pathogen earlier, respond more aggressively, and in many cases, survive the encounter that would kill an unresistant variety. The third strategy in IPM is monitoring, and this is perhaps the most human story in all of farming. Monitoring means paying attention, systematically, deliberately, walking the field not just to check progress or enjoy the growth, but to look for early signs, before the disease triangle completes itself. A leaf with an unusual color on its margins, a section of a row where the plants look slightly less vigorous than the rest, a single plant showing the first signs of asymmetric wilt. Farmers who catch disease in one plant and act immediately, removing the plant, adjusting irrigation, treating the surrounding soil, can sometimes prevent what would have been a field-wide catastrophe. Farmers who are too busy or too optimistic, who notice the early signs and tell themselves it will probably be fine, often find themselves standing in a field full of collapsed plants a week later, wondering what happened. This is not a criticism of farmers. Farming is one of the most demanding professions in the world, and the demands on a farmer's time and attention during the growing season are immense. It is simply an acknowledgement of a biological reality. Plant diseases are almost always easier to contain when caught early, and exponentially harder to address when they are not. There is an old saying in plant pathology, not a famous one, not a quotable one, just something that gets passed around among people who spend their lives studying plant disease. If you can see it, you're already late. It sounds harsh. But what it means is that by the time a disease is visible to the naked eye, the pathogen population inside or on the plant is already enormous. The infection has been underway for days. The damage is already done. What you're seeing is the aftermath, not the beginning. Now let's talk about something that is happening right now. Not in any one field, but globally. That is making the overnight death of tomato plants more common, more unpredictable, and harder to manage. Climate. The relationship between climate and plant disease is one of the most actively studied areas in modern agriculture, and the patterns that researchers are finding are deeply concerning. Phytophthora, as we discussed, thrives in warm, humid, rainy conditions. As average global temperatures rise and weather patterns become less predictable, with drier dry seasons and wetter wet seasons, the windows when Phytophthora can spread are getting longer in some regions and appearing in places where the disease was previously rare. Fusarium is also adapting. Certain strains of Fusarium oxysporum that were previously limited to tropical or subtropical climates are being detected in regions they have not historically occupied. The combination of warming temperatures and increased movement of plants and soil material around the world is facilitating the spread of strains that some local plants have never evolved defenses against. Ralstonia solanacearum, the bacterial wilt pathogen, has a known relationship with temperature. Above 35 degrees Celsius, extreme heat that is now occurring more frequently in many tomato-growing regions. The disease progresses so rapidly that even farmers who catch it early can lose plants within hours rather than days. These are not abstract statistics. For farmers whose livelihoods depend on a tomato harvest, and there are hundreds of millions of such farmers worldwide, the increasing unpredictability of disease pressure is a concrete, immediate threat. A threat that arrives in the night silently and announces itself with dead plants in the morning. Let's talk about the soil again, because there is something happening underground that we haven't yet fully addressed, something that ties together almost everything we have discussed, and that represents one of the most exciting frontiers in agricultural science. It is called the soil microbiome. The word microbiome has become familiar to many people in the context of human health, the community of microorganisms living in and on the human body, particularly in the gut. The importance of a healthy, diverse gut microbiome to human health is now well established in science, even if our understanding of all its details is still developing. The soil microbiome works on similar principles. In a healthy, undisturbed soil ecosystem, there is a remarkable diversity of organisms that exist in complex, interdependent relationships. Bacteria that fix nitrogen from the air and make it available to plants. Fungi that break down organic matter and release nutrients. Predatory nematodes that control populations of plant-damaging nematodes. Bacteriophages, viruses that infect and kill bacteria, that regulate bacterial populations. Protozoa that graze on bacteria, and in doing so, release nutrients in forms that plant roots can absorb. And crucially, beneficial organisms that directly suppress the pathogens we have been discussing. Not through any single mechanism, but through competition, antagonism, antibiotic production, and the simple fact of occupying the space and resources that a pathogen would otherwise claim. Scientists studying this suppressive soil effect, as they call it, have found that certain soils, even when deliberately inoculated with Fusarium or other pathogens, somehow fail to produce disease. The plants in these soils remain healthy even in conditions that should devastate them. When researchers analyze what is different about these soils, the answer is almost always the same: an extraordinarily rich and diverse microbial community that collectively keeps pathogens in check. This has profound implications. It means that the health of a tomato plant is not just about the tomato plant. It is about the entire invisible ecosystem surrounding it. It means that when we talk about growing healthy tomatoes, we should perhaps be talking first about building healthy soil. There is a farmer in the Netherlands, and I want to tell you about him, because his story illustrates something important. He had been growing tomatoes in greenhouse production for over 20 years, large-scale, commercial, thousands of plants in rows under glass with drip irrigation, controlled climate, optimized fertilization. By any measure, it was a sophisticated, well-managed operation. And then over a period of about 3 years, his plants began dying, not all at once, not catastrophically, but slowly, persistently, year after year. Fusarium wilt was appearing earlier in the season and spreading faster than it used to. His yield was declining. His input costs for fungicides, for replacement plants, for labor were increasing. He brought in plant pathologists. He tested his soil. He confirmed what the data suggested. His soil microbiome had been severely depleted. Decades of sterilization practices, many commercial greenhouse growers use steam or chemical treatments to kill pathogens in the growing medium, had also eliminated the beneficial organisms that would otherwise compete with and suppress those pathogens. He had created a sterile environment that was, in theory, clean. But a sterile environment is one where the first pathogen to arrive faces no competition. And pathogens, it turns out, are very good at arriving. He shifted his approach. He introduced compost with active microbial communities. He began using microbial inoculants, commercial preparations containing specific beneficial bacteria and fungi. He reduced his reliance on chemical fungicides. He planted cover crops during fallow periods. The recovery took time, several seasons, but gradually his plants became healthier. His disease pressure decreased, and when Fusarium did appear, because in a commercial operation it almost always appears eventually, it spread more slowly and his plants fought it off more effectively. His story is not unique. Versions of it are being told in agricultural communities around the world. And what they collectively point to is a rethinking of what plant health actually means. We should talk about tomatoes specifically for a moment, because there is something about tomatoes as a plant that makes them particularly vulnerable and particularly interesting to study. The tomato plant, Solanum lycopersicum, is a species that originated in South America, specifically in the Andes region of what is now Peru and Ecuador. Its wild ancestors were small, sprawling plants bearing tiny fruits, growing in a range of habitats from coastal desert to Andean foothills. They were tough plants, adapted to difficult conditions, carrying a genetic diversity that has proven invaluable to breeders. But when tomatoes were domesticated, first in Mexico by pre-Columbian civilizations, and then spread across the world by Spanish explorers in the 16th century, the selection process by nature narrowed that genetic diversity. Farmers chose the plants with the biggest fruits, the most uniform color, the best flavor, the highest yield. Over generations, the genetic diversity of cultivated tomatoes shrank dramatically compared to their wild relatives. And with that narrow gene pool came reduced resilience. Wild tomato relatives carry genes for resistance to many diseases and environmental stresses. Some cultivated tomatoes, bred for maximum fruit production, have lost some of those protective genes. This is part of why modern tomato breeding programs spend so much effort crossing commercial varieties with wild relatives, trying to reintroduce some of that original resilience without sacrificing the fruit qualities that make commercial tomatoes viable. The tomato we buy at the grocery store, perfectly round, uniformly red, with the same shape and size and color as every other tomato on the shelf, is, genetically speaking, a very different plant from its ancestors. It is optimized for production and appearance, and that optimization has costs that are measured not in the supermarket but in the field, in the soil, at night, when the pathogens arrive. Let's talk about what a farmer can actually do. Because understanding the problem is one thing, but the person with a tomato plant that is dying right now needs practical knowledge. The first step, and this cannot be emphasized enough, is diagnosis. Accurate diagnosis. Because the response to Fusarium wilt is different from the response to Phytophthora, is different from the response to Ralstonia, is different from the response to overwatering, is different from the response to a cutworm. Treating the wrong cause not only fails to solve the problem, it can make it worse. For the home gardener, several visual clues help. Fusarium typically produces yellowing that starts on one side of the plant or on lower leaves, with asymmetric wilting, and a characteristic brown or reddish discoloration visible when you slice through the stem near the base. Phytophthora produces dark, water-soaked lesions, spots that look almost bruised on the leaves and stem, often with white or grayish fuzzy growth on the undersides of leaves in humid conditions. Ralstonia produces rapid, dramatic wilt, often uniform across the plant in warm conditions, with that diagnostic bacterial ooze in water. And cutworm damage is unmistakable. A cut stem, a fallen plant, a pest in the soil. For each of these, the options vary considerably. Fusarium cannot be eliminated from the soil once established. It is there, and it is persistent. The response is prevention, resistant varieties, rotation, soil health management, and when plants show signs of infection, removing and destroying them, not composting them, because composting infected material can spread the pathogen. Never throw diseased tomato plants on a compost pile. Phytophthora can be managed with copper-based fungicides, which are one of the oldest and most widely used tools in plant disease management. They work as protectants. They don't cure an existing infection, but they prevent new spores from establishing. Applied preventively before disease pressure arrives, they are effective. Applied after the disease has entered the plant, they do very little. Ralstonia has no cure. There are no effective bactericides available for field use against it. Once a plant is infected, it must be removed. Once a soil is confirmed contaminated, management options are limited and long-term, resistant varieties, long rotations away from solanaceous crops, and in some severe cases, soil solarization, which involves covering the soil with clear plastic in summer to use solar heat to reduce pathogen populations. And overwatering. Fix your irrigation. Improve soil drainage. Let the soil dry out between waterings. In most cases, if the roots have not been completely suffocated and the plant has not also been attacked by a secondary pathogen, a tomato plant can recover from even significant overwatering stress if the cause is corrected quickly enough. There is one more thing to discuss. Something that sits at the intersection of science and history and economics. Something that connects the death of a single tomato plant in a home garden to one of the central challenges facing humanity over the next century. Food security. Tomatoes are one of the most important food crops in the world. Not the single most important. That distinction belongs to wheat, rice, and maize, the grain crops that form the caloric foundation of human civilization. But in terms of global production volume, in terms of nutritional contribution, in terms of economic value, tomatoes rank among the most significant crops humanity grows. The FAO, the Food and Agriculture Organization of the United Nations, estimates global tomato production at over 180 million metric tons annually. They are grown commercially on every inhabited continent. In countries from China and India to the United States, Italy, Turkey, and Egypt, tomatoes represent both a dietary staple and a critical source of income for millions of farming families. And the diseases we have discussed, Fusarium, Phytophthora, Ralstonia, collectively account for billions of dollars in losses every year. Not just for large commercial operations, but for the smallholder farmers who grow tomatoes on small plots of land, and who depend on those tomatoes to feed their families and sell at local markets. When a smallholder farmer in sub-Saharan Africa loses her tomato crop to Ralstonia bacterial wilt, the loss is not measured in annual revenue percentages. It is measured in whether her children eat this month. When a tomato farmer in Bangladesh loses his field to Phytophthora during the monsoon season, it is not an entry in a quarterly report. It is a catastrophe. Understanding plant disease is not an academic exercise. It is, in the most direct sense, a matter of human welfare. Scientists are working on solutions, and some of what they are developing is remarkable. Biocontrol is one of the most exciting areas. Biocontrol involves using living organisms, beneficial bacteria, fungi, or other agents to suppress plant pathogens. Rather than applying chemicals to kill a pathogen, you introduce its natural enemies. You tip the ecological balance in the plant's favor. Certain strains of Bacillus subtilis and Trichoderma, a genus of beneficial fungi, have been shown in research trials to significantly suppress fusarium in tomato crops. These are now available commercially as biocontrol products, and their use is growing rapidly as farmers look for alternatives to synthetic fungicides, and as regulatory pressures in many countries push toward reduced chemical inputs in agriculture. Bacteriophage therapy is another emerging approach for bacterial diseases like Ralstonia. Bacteriophages, viruses that infect bacteria, are extraordinarily specific. A phage that infects Ralstonia solanacearum does not affect beneficial soil bacteria, does not affect animals, does not affect the plant itself. It infects and kills Ralstonia specifically. Research into phage therapy for plant diseases is still in relatively early stages, but the results are promising, and several products are in development or early commercialization. Precision breeding, using modern genetic tools to identify and activate resistance genes in cultivated tomato varieties more quickly and more precisely than traditional breeding, is also advancing rapidly. Scientists can now identify the specific gene sequences responsible for resistance to particular Fusarium strains or Ralstonia biotypes, and use that knowledge to breed resistant varieties in years rather than the decades required by traditional methods. And perhaps most fundamentally, there is growing investment in understanding and rebuilding soil health at scale. Agricultural researchers, soil scientists, and farmers in many countries are working together to develop practices that restore microbial diversity to degraded soils, not just as an environmental goal, but as a practical tool for disease suppression. We are almost at the end of this story, but there is one more thing I want you to imagine. It is early morning, just before sunrise. In a field or a backyard garden, it doesn't matter which. The light is low, the air is cool, and the tomato plants are standing in their rows. Some of them healthy, their stems strong, their leaves catching the first light. Some of them perhaps already beginning the invisible process of their decline, colonized by something too small to see, their xylem slowly being compromised. The farmer hasn't come out yet, it is still quiet. And in the soil, a million microscopic processes are happening simultaneously. Beneficial bacteria are producing compounds that suppress fungal spores. Mycorrhizal networks are carrying water to roots that couldn't reach the moisture on their own. Nematodes are chasing bacteria through pores in the soil aggregate. And perhaps, in the soil of a field that hasn't been managed with care, where the microbial community has been depleted, and the pathogens have had time to build their populations, a Fusarium hypha is threading itself through a root cell wall, beginning its ascent. The farmer comes out, he looks at his plants, they look fine. He doesn't know yet that some of them have already lost the battle. He won't know for hours, maybe days. He won't know, in all likelihood, until it is too late to do anything for those particular plants. But here is what I want you to take from this story, and I mean this genuinely, as the true takeaway, the thing that matters beyond all the scientific names and all the disease mechanisms and all the management strategies. Plants are not passive. They are not simply things that grow. They are organisms of extraordinary complexity, engaged in constant, invisible relationships with the world around them. They communicate. They fight. They form alliances. They have been navigating a world full of threats for hundreds of millions of years, and they have developed responses to those threats that are, in many cases, more sophisticated than anything we have consciously designed. The pathogens that kill them overnight are not villains in any meaningful sense. They are also organisms, doing what their biology directs them to do, in a world that has always involved competition and predation, and the cycling of nutrients from living things back into the soil. What a farmer does, what any person who grows a plant does, is choose a side in that ancient contest. And the best farmers, the wisest farmers, the ones whose plants survive year after year in the face of all this invisible pressure, are not the ones who wage the most aggressive war against pathogens. They are the ones who best understand the ecosystem they are a part of, and who work to make it healthy enough to fight its own battles. The next time you pick up a tomato at a market, smooth, red, warm in your hand, take a moment to think about the journey that tomato made. Not the journey in the truck or the journey from the field to the warehouse. The earlier journey, the invisible one. The one that happened in the dark, in the soil, in the quiet hours before dawn, when something very small was trying to end what was being grown. And the plant, and the farmer, and the ecosystem, and all the accumulated knowledge of generations of people who learned what we have just talked about, found a way to survive. Every tomato that reaches you is a small victory. Fought not with drama or noise, but with chemistry, with biology, with care, with knowledge, and with the remarkable resilience of a plant that has been feeding human beings for 500 years and is not done yet.